Right, well, I think we should make a start. So welcome to everybody who's joined us for this HDBI ethics seminar. So you'll remember this is the Human Developmental Biology Initiative, Wellcome funded multi-center program of research in human development. And this is ethics seminar number 7. So we've had a very informative and enjoyable series of ethics seminars. I'm sure today will be no exception to that. Just to say that those of you who've joined before, we always say that some of the topics that are dealt with in these sessions can be contentious, and some people hold very strong views. We hope that after the two speakers have given their talks and we have the Q&A, that we'll be able to have an open, inclusive discussion, and we really encourage everybody to participate in that. Pilar has put details of the Slido login, so you can put questions there if you want, and she'll have a look at that account when we get to the Q&A. Or you could write things in the chat, or of course, when we get to the Q&A time, you can put your hand up, and I'll go through people in order to ask their questions or make their points. We're recording this session solely the speakers' presentations. Then we will turn off the recording and not record the actual discussion period. So I'm delighted to have two speakers here today, Stephen Wilkinson and Hank Greely. They'll talk in sequence, and then we'll have the Q&A after Hank's presentation. So our first speaker is Professor Stephen Wilkinson from Lancaster University. Stephen is a distinguished professor of bioethics, and he's been involved in this kind of research for a number of years now. So he, together with Rosamund Scott at King's, he led a Wellcome funded research program for 8 years, 2013 to 2021, on the ethics and regulation of human reproductive donation. And he's now involved with another Wellcome project on the future of human reproduction. He works with colleagues in a variety of different fields, so design, English literature, law, linguistics, philosophy, psychology, and sociology, so very multidisciplinary. Last year, Stephen was appointed to Nuffield Council on Bioethics, which many of us have interacted with, and which does wonderful work in this area of ethical issues arising from ongoing research. And Stephen was a member of the Council's working group on the stem cell-based embryo models, which published a report in November last year. So thank you very much, Stephen, for agreeing to speak to us today. Title is Human Stem Cell-Based Embryo Models. What are the ethical issues? Over to you.
Okay, thank you very much. So let me just share my slides. Right, you can see that. I've actually changed the title to What are the Issues, because I realised that they're not all ethical issues. Thank you for that lovely introduction. Right, so anyway, thanks for the introduction. The first couple of things I was going to say have possibly already been covered to some extent. So what I'm planning to do today is give you a few highlights from the Nuffield Council on Bioethics report from November last year, called Human Stem Cell-Based Embryo Models Review of Ethical and Governance Questions. So that's just kind of me telling you what it says, really. I will then also, so along the way, weave in a few sort of broader points and some analysis of the conceptual and ethical issues raised by these entities. As Andrew has already mentioned, I should put this on the table for sort of disclosure. I am a member of the Nuffield Council on Bioethics. I was a member of the working group behind the report. Today is not an official view, it's a personal view. I mean, I'm happy to defend what's in the report and so on, but I'm trying to sort of pick up on the more general issues and themes raised in the report. I'm not speaking in a sort of official capacity. And as Andrew also said, I should also say that I'm also part of a Wellcome funded project called the Future of Human Reproduction, which looks at new reproductive biotechnologies from a sort of humanities and social science perspective, and tries to think about new ways that we can think about those. So that's just putting all my disclosures on the table at the beginning. So to start, What are stem cell-based embryo models? Well, the other thing I should say before getting going is what my background is. I'm a philosopher really by training, and bioethics is my area. So obviously I don't claim any kind of scientific authority. I'm not a biological scientist. I'm not any sort of scientist. So there's lots of people in the audience who probably know more about the detail of this than I do. But in my understanding, and this is what it says in the report, is that stem cell-based embryo models is an umbrella term for a range of structures created from stem cells which resemble or replicate aspects of embryonic development. These stem cells are derived either from embryos, embryonic stem cells, or through the reprogramming of cells from other human tissue, such as skin or blood cells, induced pluripotent stem cells. While a number of different terms are used to describe these structures, we've adopted the term stem cell-based embryo model, recognizing the value of consistency with other formal guidelines and frameworks. So one other thing to say at the start is I think I'm going to, in this talk, it's a difficult thing to talk about. Nobody can say SCBEMs. So there's two things I'm going to call them. I'll call them embryo models. That's nice and easy using normal words. And I might also call them SCEMs, because in the working group, we did have this discussion about whether you could pronounce SCBEMs. So I'll say SCEMs and I'll say embryo models. And they mean the same thing when they come out of my mouth in this talk. But yes, the B is a bit problematic in terms of making that a catchy acronym. So that's what they are. Probably, again, it's worth thinking about terminology, because this often shapes the way people think about things. So some other things that they have been called are artificial embryos, synthetic embryos, embryoids. There's another acronym called sheaths, which I've written out on the screen there, synthetic embryos with embryo-like features. These are all terms that have been, there's probably some other terms out there as well. The working group view was very much that the key thing was to be understood and to interact with other existing work by other committees and other academic research, and SCBEMs seem to be becoming, the most commonly used expression. So that was what was used, like not necessarily because it's the best term in absolute terms, but it just fits in with existing uses in other places. And as I'll come on to say in a minute, I think the idea of model is perhaps slightly tricky in this area, or at least from a kind of public understanding of science point of view, might be a bit tricky. A key point about stem cell-based embryo models is that some of them are a lot more like embryos than others. So as I understand it from hearing various talks and bits of evidence on this, some of them are more like sort of stem cell lines that are trying to model particular tissue types or parts of the body, and they're not whole things at all. They're just sort of modeling a particular type of embryonic tissue. And so those entities are a lot unlike embryos. They're not like embryos at all. And there's just no way they could ever become have any reproductive potential, and that was one end of the spectrum. At the other end, there were some entities, or at least could be some entities, which are a lot like embryos morphologically, functionally, and so on. And views vary on this, but people have claimed that ultimately there is a possibility that those things could have reproductive potential, although that's as yet completely unconfirmed, and would require, obviously that would be a long way down the track scientifically, and also would require a lot of ethical and legal debate before we got anywhere near that. But that's the wide range. You've got sort of individual tissue cell lines, tissue lines, ranging right the way through to things that could in principle look a lot like an embryo. So that's one of the complexities of thinking about this as a unitary topic ethically, or from a government's perspective. You've just got a really wide range of entities that you're thinking about, and maybe in due course the way we categorize things will become more nuanced, and there'll be this term SCEMs will disappear completely, and there'll be 3 other terms for 3 different types of things, or 10 or 100 things, whatever it is. Maybe the categorization landscape could change quite quickly. On model, I mean, the reason why I'm always a bit nervous about, I think I only heard of stem cell based embryo models quite recently. I mean, it's maybe 2 to 3 years ago. It was quite new to me, I actually, like a lot of people hear this expression for the first time, I think, was struggling to think, what is that? So literally the first time I heard that expression, I was thinking, is this some kind of simulation, representation? Is it a framework? Because the word model can be used in lots of different ways. And I think when you talked when but people ask you what you're doing, and I say, well, I'm on this working party on stem cell-based embryo models. People who are completely outside the field. More than half of those people, I think, were quite surprised that a stem cell-based embryo model is physically quite a lot like an embryo. I think they thought it was going to be something else. So there are lots of senses of the term model. I mean, I've put a few. I mean, some of them are completely irrelevant. So you've got fashion models, and you get models of phones and cars, and you could try to be a model colleague, meaning you're exemplary. So those are all, put those all to one side. But there's a few other senses of model which are in the same general ballpark. So you've got, models can be representations, they can be frameworks, they can be simulations. So I think that's what throws people when you say you're looking at embryo models. They're almost thinking you might be looking at some kind of simulation or theoretical framework or something. And that's, obviously you can explain what's going on, and then that's fine. But I think from a public understanding point of view, I think there is a bit of a barrier set up by the term model. And some of the events I've been in with members of the public, they've struggled a bit with that, and they say, well, like, why is it a model? Why isn't it an embryo? Anyway, so what we're talking about here is a physical model, which is a perfectly reasonable sense of the word model. And some other examples of this use of the word model would be model aeroplane, model car, model village. They're physical things that are like the thing in some respect, but not so much like it that they are the thing. And in particular, I think for physical models to be models rather than the thing itself, they have to sort of differ in a couple of ways. I mean, normally sometimes they're a different size. So model aeroplanes are typically a lot smaller than real aeroplanes. And they also tend to lack important functional features. So in a model aeroplane, I mean, if you could put 8 passengers on a model aeroplane and fly it across the Atlantic, that would just be an aeroplane. It wouldn't be a model. So I think those are the key things that stop something from being the thing itself and make it a model. And that's kind of what's going on here. I should say that maybe A on my slide is parasitic on B. So it might just be the function that's important. I mean, perhaps size is just the thing that stops it from having a function. But anyway, I mean, this might seem to be a bit round the houses, but I think it's worth thinking about why is an embryo model and not an embryo? And ultimately, it boils down to this, that it lacks important functional properties. It isn't actually a different size. I mean, size isn't really so relevant here. It's just about function. So it could only really be a model, I think, if it can't do everything an embryo can do. And that's what happens with model aeroplanes and model cars and so on. So if an embryo model, or model inverted commas, became very similar to an embryo, then I think there's at least a question about whether it would be an embryo. So it's worth bearing that in mind as we go. I don't think it's a hard distinction. The other thing to say, and again, I'm probably getting overly pedantic about the meaning of model here, but in a lot of domains, I think the boundary between being a model of something and being the thing itself is actually a little bit vague. So I mentioned lightheartedly the aeroplane example, but I mean, those kind of transport examples probably are quite telling. So I'm told, it's not my hobby or anything, but I'm told that there are large model railway things that can carry a passenger, but they only go a short distance. So, that's sort of moving into the borderline territory, isn't it? It's got some of the functionality of a rail system, probably a lot of the functionality of the British rail system, and and you can imagine getting a bigger and bigger train and still saying, well, that's definitely a proper train. No, it's not a model. So and probably it sounds a bit trite as an example, but probably embryo models are like that in the sense that this continuum of function, and at some point it will tip into being an actual embryo. That's my view. That's not sort of in the report. So you might want to know why should we use SCBEMs? And there's sort of 3 big reasons, and then on the slide, so it's so small, a list of sort of examples are given in the report of ways it could be used. So one of the big advantages of stem cell-based embryo models is that it would provide you with a sort of plentiful supply of research, or could provide you with a plentiful supply of research material compared to having to use actual embryos. So in terms of the scale and effectiveness and cost effectiveness of science, it could give you a really useful research tool. The second, so that's just about quantity really of material and how hard it is to get embryos for research. The second one's more about regulation and perhaps possibly ethics. And the thought is that because of the 14-day rule, which I'll say a bit more about in a minute, we can't currently research on embryos 14 days after fertilization. And there's a period which some people call the sort of black box period, which is 14 to 28 days, when a lot of really interesting things happen developmentally to human embryos. But it's very, very hard to study it because the 14-day rule prevents embryo research from going beyond that point, and it's hard to get material from other sources to study that. So there's this sort of, so we don't know as much as we could and should about that period, and stem cell based embryo models would enable you to, or could enable you to research on them instead during that period, and enable you to find out things about embryonic development. I mean, obviously there's a question about how closely they resemble embryos, which you can't really answer definitively without doing the research on embryos, but you know, the thought is you could get an indicative set of data about that if you got the models right. So again, very scientifically advantageous, and currently there's a block on that due to the 14-day rule. Finally, this is the point at the bottom. So the Nuffield report does stress that SCBEMs research will complement rather than replace embryo research, and it goes out of its way to say there's not necessarily anything bad about embryo research, and we shouldn't be trying to completely get rid of it. But some other people do say, and it's often said that SCBEMs research will, reduce the amount of embryo research, or could ultimately reduce the amount of embryo research that there is. And so if you're someone who does have concerns about embryo research, then that's a positive for you. So that's an argument that's sometimes put out there. There's a long list of possible things we could do with SCBEMs research. So it might help with IVF success rates, developing new forms of contraception, enable us to develop culture media for embryos, could potentially be used in training or to improve techniques like freezing or thawing embryos. And further down the line, and perhaps more controversially, we've got things like creating tissue for transplantation, and eventually maybe even reproductive use if it's deemed ethical and safe. But as I said earlier, that's a long way down the tracks and would have to be after a considerable amount of ethical and public debate. All right, so I mentioned the 14-day rule. So this is a bit of a side point really, but probably worth you knowing. So the 14-day rule, as I said, prohibits embryo research after 14 days after fertilization. It's a sort of widely accepted sort of ethical convention, and in some countries it's also part of the law, including the UK. But recently there's been some move away from that from a number of notable bodies. So the three examples, I won't read them out because I'm already becoming slightly conscious of time, is the ISSCR removed its support for the 14-day rule in its 2021 guidelines, suggesting that there should be public and societal debate about this. So they didn't sort of recommend getting rid of it, but they stopped supporting it, and said it would be worth looking at. The Health Council of the Netherlands in 2023 recommended that the 14-day rule be extended to 28 days. And most recently, and most interestingly for us, the HVA very recently in November 24 recommended that the laws change to extend the time limit on embryo research. And they say we've seen significant advances since the law was introduced in 1990, and it's now increasingly possible for researchers to develop and sustain embryos beyond 14 days. And this could provide valuable information in a strictly regulated environment for the kind of reasons I just gave with the black box period and so on. Right. So now I'll now give you a quick summary of some of the key recommendations in the report following that preamble. The first one is that is support for existing proposals. So you may or may not know that in 2024, a UK code of practice was produced by Cambridge Reproduction and Progress Educational Trust that sets out standards for SCBEM research and recommends the establishment of an oversight committee and a register to review and record SCBEM research. So the Nuffield Committee supports that proposal and thinks that it's a good proportionate first step. Obviously, it won't do the job on a permanent basis, but it's a very good first step, and would give us a sort of, if you like, a dedicated oversight committee, which is in some respects akin to an ethics committee, or would oversee ethics committees that looked at this very specialist area of research. That committee doesn't yet exist, but the Nuffield Council report supports it as a sort of good first step, and in particular thinking how long it can take to do things like proper legislative reform, for example, is something that could be done, you know, relatively quickly, and so that's a good first step. So totally on board with that. Secondly, what sort of legal status should embryo models have? So the report makes clear that its view is that stem cell-based embryo models and embryos should have quite distinct legal and regulatory statuses, and that this should be reflected in the Human Fertilization and Embryology Act. And for governance purposes, the stem cell-based embryo model should be considered to be distinct from an embryo, even if a time may come when, for scientific purposes, the two could become indistinguishable in terms of their capacity for forward development. So to just try and unpack that slightly, so there's different ways you could approach this. I mean, one way of approaching it would be to look at SCBEMs on almost a case-by-case basis, perhaps try and give some characterization of what features something has to have in order for it to count as embryo. And perhaps that would be functional, perhaps it would be to do with reproductive potential, or there might be some sort of biological proxies for that. And then for each SCBEM, you would have to do a sort of case-by-case assessment of whether it was an embryo or not. Now, as you can imagine, that's potentially really, really complicated and difficult, and requires judgment calls that even a very well-informed, well-meaning, conscientious person might find quite hard. Some of them could be easier. So I mentioned earlier that some SCBEMs are nothing like embryos, and perhaps that's easy. But in the middle and at the other end of the continuum, there could easily be SCBEMs which, you know, we just don't know. And so the downside of that approach is it will put either scientists themselves or possibly sort of these ethics committees that are overseeing them in quite a difficult position of having to keep judging what the status of the entities is. So that's a possible approach, but the committee thought it would be much better to have a distinct category, and so the argument for that is here. So it's really a pragmatic argument. The thought is, as I've suggested, it would be very, very hard to have a situation where some SCBEMs were treated as embryos and some weren't. So, as it says in this quote, differential treatment in laws based on the pragmatic ground that governance as a separate entity will be safer and more proportionate. So the problem with the alternative approach is it's messy, it's costly, it gives researchers a lack of certainty. And whereas this, we think, is going to be a much clearer sort of position, provides clarity and certainty for researchers. It prevents, or at least reduces the risk of courts having to deal with very difficult cases and then potentially perhaps overreacting by bringing within the definition of embryo too many things. And so the thought was this would be a much clearer approach, and it will also provide has the potential to provide the public with reassurance, particularly when combined with some of the red lines, which I'll talk about in a minute. Right, I'll do that now. Red lines. Actually, the more I say this, the more I think red lines is a horrible cliche, so apologies for that. I should call it something else. But anyway, the first one in the report is that well, I'll read it out. It says, our proposed amendment to exclude SCBEMs from the definition of embryo should be made in conjunction with a prohibition on transferring a human SCBEM to the in vivo reproductive tract of a human or non-human animal. So this is a very important sort of reassurance, and the thought would be that it would be illegal to implant a SCBEM into, well, as I said, the reproductive tract of a human or non-human animal. So that's sort of completely blocking, in a very sort of hard law way, use of SCBEMs for reproductive purposes. I mean, as by way of an aside, I should say, I mean, I can imagine a world where one of these instances wouldn't necessarily be unethical. So you can imagine a long way down the line, if the technology came, if the science got to the point where we knew that the SCBEMs were going to be reproductively safe and as effective as embryos, then in principle, I'm not sure personally that I would support that forevermore as an ethical prescription, but as a sort of, as this recommendation, as a sort of the here and now, real world legal prohibition, that seems absolutely sensible to me. So that was the first one. The second one's about time or stage limits, and as I haven't said yet, but will now, the 14-day rule doesn't really work very well for stem cell based embryo models for a number of reasons. One of them is there's no point of fertilization, so it's hard to know when the clock starts. A second one is that the pace of development can be very different from embryos, so I'm told. So some SCBEMs develop much more quickly, or in a different or with different developmental stages from an embryo. And some of the entities sort of can be created in a way which means they sort of come into existence at a much later developmental stage than conception. So a pure time limit doesn't make too much sense. So what at the moment, the proposal is the oversight committee sets a case-by-case time limit for each individual project, and the Nuffield report is supportive of that. But on top of that, the key suggestion is the one here, which is that no embryo model should be developed with features consistent with Carnegie stage 23, which in a regular embryo is 56 to 60 days or beyond, if it has the potential to develop capacities for pain and awareness, or if the purpose of the research is to explore the feasibility of reproductive purposes, or if the model has been genetically altered to avoid capacities referred to in 1 or 2, resulting in a model that would diminish public confidence in research if the model were to proceed beyond this threshold. So as you can see, and I won't have time to go into unpack all of the detail of this, but it's there for you to look at. It's not a hard time limit. The thought is that you're taking these features and you're taking the features in Carnegie stage 23, which is it's a bundle of features that normal embryos have at this point in time. And you're saying that you mustn't go beyond that with stem cell based embryos if, excuse me, if one of those 3 conditions is met: pain or awareness, feasibility of reproductive purposes, or if there's been some attempt to sort of bypass that by genetic modification. Now, the argument for this view is partly about, it's largely about arbitrariness. I think the thought was that this is a less arbitrary point than some other earlier ones, because in particular, this represents the point at which embryos become fetuses, so where they become classified as fetuses, and a lot of the developmental work that takes place in embryos happened by that point. Now, it's important to say that this isn't, first of all, this isn't an ethical boundary, it's a practical boundary. And second of all, it's an absolute top limit. So you'd have an oversight for each project that comes along, you'd have an oversight committee that sets for that project a time limit. And that time limit would be based on a blend of ethical considerations and also what the scientific justification was for carrying on for that long in time. So the oversight committee can set a much lower limit for any individual project, but the proposal is that this is the sort of absolute maximum which we have if those conditions are met. So it sort of overlays that on it. So this is thought to be better than not having any limit at all, and it's the least arbitrary version. And it may be that later on there has to be a lower limit or a different limit, but that's something that needs to be done, in the context of a wider sort of public debate. I'm going to skip through the 3-stage approach to regulation because I'm running out of time a little bit. So I will just summarize the main proposals, give you a couple of reflections, and then that's that. So key proposals in the report: embryo models shouldn't be regulated as embryos. There should be a distinct legal category. There should be a ban on reproductive implantation in humans or in other animals. There should be an upper limit. It's not a time limit, but it's based on Carnegie stage 23, which is a developmental stage that embryos, typical embryos, have around 56 to 60 days, and that's in conjunction with potentially lower limits for individual projects set by an oversight committee. And as I skipped over, there should be a 3-stage evolution of a regulatory system, which starts with the voluntary code, moves through primary legislation and regulatory sandboxes, and ends up with a statutory committee dealing with at least a subset of stem cell based embryo models. So some final reflections, which won't take too long. I mean, first of all, as I alluded to earlier, the administrative and legal categories in this area don't always necessarily need to reflect what I call there the sort of ontological or scientific reality. I mean, by ontological, I mean kind of what categories of things there really are in nature. So it may be that sort of ontologically, philosophically, scientifically, some SCBEMs are embryos, But from a kind of administrative and legal point of view, I do think it's defensible and sensible to keep them apart. So, and there's lots of, I haven't got time to give you any examples, but we can think of lots of examples where the legal version of the concept isn't quite the same as the scientific or the philosophical version for reasons of efficiency and clarity and so on. Yeah, we could try to mess with the definition of embryo to sort of deal with this problem, but I think most of the ways of doing that will probably be quite complicated or unmotivated. So you could, for example, say that something's only an embryo if it's the result of fertilization. You could say that proper embryos have got some kind of dignity or symbolic value that SCBEMs don't have, but that seems question begging, because I don't see why somebody couldn't, if they wanted to, attach symbolic value and dignity to SCBEMs if they're very embryo-like. And you could also talk about, what's the intention of creating these entities, but again, that seems insufficient, because embryos could be created for different purposes, or a SCBEM and an embryo could be created for the very same purpose. So those things, I think, are not going to work. So I think the approach proposed of keeping the administrative and legal categories distinct from the underlying ontological scientific reality is probably a good one, a sensible sort of pragmatic solution. So I've given you quite a lot to think about there. Thanks for the invite. I'm happy to take questions later at the end, but otherwise, thank you very much.
Thank you, Stephen. That was great. And as you say, it's given everybody a lot to think about. So we will come back to you with questions after Hank's talk. So now we'll move on to Hank Greely, our second speaker. Hank is the Dean F. and Kate Edelman Johnson Professor of Law, and also Professor by Courtesy, I love that title, Professor by Courtesy of Genetics at Stanford University. I hasten to add that Hank is not sitting up in the middle of the night in California. He's actually visiting Spain at the moment, so he's on a more reasonable time frame for this presentation. He has specialized for many years in ethical, legal, and social issues arising from advances in the biosciences. He's been particularly interested in genetics, neuroscience, human stem cell research, and he chairs the steering committee of Stanford University Center for Biomedical Ethics, and also Stanford Center for Law and Biosciences. So we're really looking forward to your presentation, Hank. The title is Human In Vitro Gametogenesis and Its Possible Implications. So thank you for joining us, and over to you.
My pleasure. Thank you for inviting me. So first, I have no slides. I know that's practically unheard of in the scientific field. It's never been clear to me whether it's against my religion to use slides or whether I'm allergic and it would cause an anaphylactic shock. But either way, whether it's damnation or whether it's instant death, I've just never been willing to take the risk. So I am a talking head. I do want to talk about in vitro gametogenesis, and specifically I want to talk about 4 things. First, how we would go about making gametes. Second, why we would go about making gametes. Third, the issues, ethical, legal, practical, political, and otherwise, that in vitro gametogenesis would raise. And then fourth, some thoughts on what I think should be done. I will say that this topic first came into my mind in October 2010, so 15 years, almost 15 years ago. I was at a stem cell conference in Münster, Germany, and one of my colleagues, a religious studies professor from University of Chicago, then at Northwestern, named Lori Zoloth, was talking about these new, exciting, induced pluripotent stem cells and all the things they could do. And she said, it was one sentence, and what if you could use them to make gametes? And I thought, what if you could use them to make gametes? What would that mean? And I thought, I need to think about this some more. And then I concluded, well, maybe I need to write something about this. It took me about 6 months to write a book about it, although it took me 6 years to find the 6 months in which I wrote the book. But in 2016, I published a book called The End of Sex and the Future of Human Reproduction. It's now closing in on 9 years old. but I still think it's a pretty good reflection of my current thinking about where things are going to go. So I'm not giving you any slides, but The End of Sex and the Future of Human Reproduction available at bookstores everywhere as long as they're called Amazon. So let's go first, how to make gametes. Well, one thing is, what are gametes? I think this audience knows that. One thing I hadn't realized until recently was that the word gamete actually comes from the Proto-Indo-European verb to marry. It was turned into a term for eggs and sperm in 1878 by a German scientist named Eduard Strasburger, and it's been used that way ever since. Almost all of us have made, are making, or will make gametes. I suspect that there are no prepubescent males in this audience, so probably there's nobody who's going to make gametes who hasn't been making gametes yet, but there are a lot of them in the population. One of the things that hurt me most about writing The End of Sex was the chapter I had to slice up most bitterly, most strenuously, because it was too long and too complicated, was the chapter on how human reproduction works naturally, which is weirder than I had ever imagined. How gametes are made, when gametes are made, where gametes are made is truly amazing. Extra embryonic tissue, yolk sac, migrations, partial meiosis before birth for eggs, all sorts of weird stuff. All I'm going to say about it is, for most of us, it works. We make gametes, so we know gametes can be made. The question is, how could you make them in vitro? Now, presumably you could use embryonic stem cells. Most of the attention, for reasons that I think will become clear, has been focused on induced pluripotent stem cells, because that way we could take cells from Andrew, and we could, from his skin, we could take fibroblasts from Andrew, and we could make sperm from Andrew that would have half of Andrew's DNA. Or we could make eggs from Pilar that would have half of Pilar's DNA. Because induced pluripotent stem cells, if we're using the person who intends to be the parent as the source of the donor cells that become the induced pluripotent stem cell, they've got that genome. Whereas if you're using human embryonic stem cells, you've got embryonic cells that are from a variety of different parents. So you could use ESCs. Most of the intention is on iPSCs. So far, it has clearly worked in mice. Now, of course, we all know that the way scientists will eventually cure all humans of all diseases is figure out a way to turn us all into mice, because we can cure everything in mice. And one of the things that I've learned by studying reproduction is often very oddly species specific. I mean, mice and rats are pretty close. Mice were cloned very quickly. Rats, it took forever. Dogs and cats, pretty close. Cats were cloned quickly, dogs very, very, very slowly. So the fact that it's been done in mice is evidence that it can be done. It is not very strong evidence that it can be done in humans, but it seems to show that there's no general block. In theory, what you do is take these induced pluripotent stem cells, turn them into, in a first step, primordial germ cells. This is something that happens when we're embryos, when we were embryos. Probably shouldn't be allowed to happen if we were embryo models, but that's Stephen's talk, not mine. So when we were embryos early on, we formed these primordial germ cells. The primordial germ cells are not yet differentiated as to whether they're going to become eggs or sperm. It is only after the primordial germ cells then migrate into the tissues that will become either testes or ovaries that the second stage takes place, where they differentiate into either eggs or proto-eggs, pre-eggs, or sperm, pre-sperm, in these embryonic and then fetal gonads. In theory, what people are trying to do, and what's worked with mice, is basically take iPSCs and expose them to cells and sort of quasi organs that replicate the environment in which, in nature, these pluripotent stem cells become first primordial germ cells and then either eggs or sperm. The best results have come from two labs in Japan, that of Mitinori Saito and that of Katsuhiko Hayashi. Hayashi was originally Saito's student, but they now both have labs at different universities. Back as long as 12 years ago, they were able to make living, healthy mice from stem cells that had been turned into eggs and stem cells that had been turned into sperm. They weren't able to do it at a high level of efficacy. It wasn't very efficient. It was about 1/20th as efficient as using actual eggs and sperm from mice, but they were able to do it. And they weren't able to do it with complete success. There were, even the ones that were born alive, often had various disabling conditions. Over the years, they've worked on it, and I think things are getting better, but it's still, even in mice, not very efficient and not particularly safe, but it has been done. In 2023, they took another step and were able to take cells from a male mouse and, in a nice tricky way involving waiting for Y chromosomes to drop out as cell lines propagated, turn them into eggs and have live-born mice that had, quote, two fathers, close quote. One provided the sperm, one provided the induced pluripotent stem cells that became eggs, and the result was actually some mice. So with mice, good progress is being made. Problem is, we don't care that much about mice. In humans, there has been a great deal of interest. There has been a little bit of work in non-humans, particularly some interesting work in cattle, but most of the attention has focused on humans, and there the work is has progressed not nearly as far. People are not mice. Primordial germ cells have been produced, or things that look a lot like primordial germ cells. Some of them have been taken some of the steps from primordial germ cell to mature oocyte or to mature sperm. But no one, as far as I know, no one has announced that they've been able to produce a mature human egg or a mature human sperm using iPSCs yet. That could change tomorrow. There are currently at least 5 different labs around the world. And when I say at least 5 different labs, I say that with some sorrow. I wish there were more. But I know of 5 in particular, the 2 labs in Japan. Azim Surani at Cambridge has been doing this kind of work. Last year, he published a paper on making human primordial germ cell-like cells from pluripotent cells. Amanda Clark at UCLA has been doing this. And there's a Chinese group that has been publishing on these issues. I've not been quite clear on what exactly they're doing, but they had a recent paper on how they were able to make, I believe it was eggs, by changing the epigenetics, making 20 different CRISPR edits in a cell line that may turn out to be interesting. There's also something going on in another forum. Back in about 2023, IVG had its moment in the venture capital sunlight, and at least 4 different companies were formed in or around 2023 with the goal of making in vitro gametogenesis in humans. Conception Biosciences got the most publicity. They had a very publicity-eager founder who is a gay male who wants eventually to be able to have genetic children who are half his genes and half his male partner's genes. Ivy Natal is another company. Vitro Labs is a third, and there is one of the 4,619 spinoff companies from George Church at Harvard is called Gamito, which was also aimed at this goal, although recently they've been moving more into ex vivo oocyte maturation, and they had an interesting paper on that published within the last couple of months. So nobody has gotten to the proof of principle stage with humans yet. People are trying. There's not a lot of public research funding support for this kind of work. And not that there's likely to be any public research funding support for any kind of bio work in my country anytime soon. We'll see. We live in hope and fear. But reproductive stuff does not get a lot of funding for various political reasons. But there is some venture capital funding coming in, and there are some labs that have been able to get some funding and do some work. So it has gotten to the point where recently the HFEA held a workshop on this and concluded that this is something that is plausible enough that could well happen within the next 10 years, that they think more attention needs to be paid to it. A conclusion in which I heartily agree. But it's nice to have my view joined by the HFEA. So second, why do it? The biggest reason to do this is infertility that's caused by people not having gametes or having so few or such disabled gametes that they can't have children. Now, there are many ways for people to be infertile. Many of them have nothing to do with gamete production. I mean, traditionally, things like blocked fallopian tubes or difficulties in ejaculation led to infertility. Those are things that IVF can deal with, in vitro fertilization can deal with. IVF can't deal directly with the lack of gametes. I mean, it can do it indirectly by using donor gametes, something Stephen's written quite a lot about, but it can't make new gametes for you. So if you are a male who, say, had mumps when you were 12 years old, there's a good chance that you don't make sperm. If you were somebody who had cancer treatment in your adolescence or early adulthood, there's a good chance that you don't have any functional gametes. If you are somebody who was born with a particular condition that rendered you agametous, you can't have biological, genetically related children unless IVG works. There are no good numbers on how many such people there are. I know some of them. I believe that there are hundreds of thousands of couples in the United States, perhaps as many as several million, who fall into this category. Multiply that 20-fold by the proportion of the US population compared to the proportion of the world's population. You get a lot of people who cannot have genetic children. Some of them don't care. Some of them are happy not to have children. Some of them aren't happy, but if it's God's will or Darwin's will or whoever's will, they'll live with it. Some people get desperately unhappy. This is a source of great suffering. It's not physical suffering, but I don't think suffering has to be physical. In fact, I think the worst suffering is not physical. There are people who go through 5 and 10 rounds of IVF at great expense and great physical and emotional trauma in a desperate effort to do something that they can't do. So I think the best reason for IVG is to let people who desperately want to have genetic children, quote, of their own, close quote. And I put that in scare quotes because, frankly, I think providing the sperm is the least important thing about being a father, that adoptive parents are true parents. But there are people who really want, for various reasons, to have a genetically related child, and they suffer. There are other issues. Egg harvest is the hardest, most expensive, and most only really dangerous and risky part of IVF. It's one of those areas where life is unfair. Sperm harvest is rarely unpleasant, difficult, or risky. Egg harvest is always unpleasant, difficult, risky, and expensive. You could get more eggs for research purposes, and then you could use them for expanding pre-implantation genetic testing, something I will come back to. Third point, the consequences, the issues raised by IVG. First, and this is most important, we tend not to think of this as an ethical issue, but it is for me always the most important ethical issue, safety. It is fundamentally unethical to do things unreasonably unsafe to people. And that's particularly true, I think, when the people you are doing something unsafe to are people who've never been able to consent. The parents were able to consent, but babies born who might have serious, serious problems because of being born through IVG, they never had a chance to consent. I don't think that's a stop. I mean, I don't remember the consent form I signed saying, yeah, I'm willing to be born, and particularly to these people. And we do things that affect early development embryos and fetuses all the time because we have to. But I do think it makes safety even more important. So the biggest ethical issue is it safe? And I think that'll take at least 10 years of preclinical and clinical work before you can mainly 10 years of probably preclinical work with non-human primates, as well as other species, before you can even get to a phase 1 human clinical trial. But if things went well, I think you could get to a phase 1 trial within 10 years. and then work your way up and see how safe it is and isn't. Then you get to a variety of really sometimes interesting, sometimes bizarre IVG specific items, like if we could make eggs or sperm from skin cells, well, 90-year-olds have skin cells. Why not make eggs from 90-year-olds? Sperm are being made by 90-year-olds, at least 90-year-olds, some 90-year-olds. But eggs aren't being made by 90-year-olds. Eggs aren't being made usually by 50-year-olds. In fact, almost all eggs seem to have been made before a female child is even born. They were made in utero. If you could make more eggs, there are a lot of people who would be older who can't make eggs anymore or don't have any useful eggs anymore who might want to do this. But do we have concerns about 90-year-old genetic mothers? Do we have concerns about 9-year-old genetic mothers? Do we have concerns about making people genetic parents who have been dead? You may remember that Dolly, the famous sheep, the world's most famous sheep, was cloned from a cell line made from a ewe that had died 3 years before Dolly was born. Freeze cells well, turn them into iPSCs, thaw them, turn them into iPSCs, turn them into gametes. You can have dead parents. Now, of course, we've had dead fathers who died after conception but before birth, but this could look very different. What about this one? It's hard to steal eggs. It's really hard. It's hard to steal sperm. It's really hard to steal eggs. It's not that hard to steal somebody's cells. If Andrew has a paper coffee cup and he throws it away, I'm sure he doesn't. I'm sure he's more environmentally responsible than to use paper coffee cups. But if he throws it away, there are probably some living cells from Andrew on that coffee cup. And maybe if I picked it up, I could make him a dad without him ever knowing it. Now, of course, there would be a big commercial market for Andrew's sperm, I'm sure. But if we made him a famous soccer player, football player, there'd be an even bigger market, I suspect. Mbappe, people should follow Mbappe, who just had a hat trick against Manchester City yet last night here in Madrid, where I am. The Bernabeu Stadium is 2 blocks from where I'm living. I heard the crowd. His sperm would, I'm sure, command a big premium, especially if he didn't know anything about it. What about this one? If we can make eggs from men or sperm from women, and why would we want to do that? Well, one of those classes of people who don't have the egg and sperm are same-sex partners. They may both make gametes, but they both make the same gamete, and you need opposite gametes. If we could make eggs from men or sperm from women, that fixes the fertility problems of gay couples. But what if we took Pilar's fibroblasts and made eggs, and Pilar's fibroblasts and made sperm, and made Pilar embryos and transferred them into Pilar's uterus, and 9 months later, Pilar gave birth to what exactly? I called it a unibaby. Note, it is not a clone. For every genetic locus where Pilar is homozygous, it's going to be the same. But where she's heterozygous, let's say Pilar's blood type is type A, and she's a heterozygote of A and O. You get to the classic Mendelian distribution. There's a 1/4 chance that the unibaby would be AA, a 1/4 chance it would be OO, and a 50-50 chance it would be AO. So it's not a clone. It's closer than a SIB. What is that? Or let's take a couple, we'll go back to couples. We make an embryo. We kill it, use the HESCs to make another embryo. Meanwhile, we've taken some other couples. We've taken 16 couples, and we make embryos from each of them. And from the embryos we make from each of them, we make eggs and sperm, and we keep crossing and crossing and crossing until you've got one embryo that has 32 parents. Multiplex parenting. Now, in a way, all of us have 32 parents. You just have to go back to great-great-grandparents, and we've got 32 genetic ancestors, probably, although one of them may have dropped out at that point, thanks to recombination. But that's kind of weird. And then, of course, a very different IVG, but important ethical issue, at least in my country, is access. Is everybody going to get this? Almost certainly not. at least not at first. It'll be rich people only, people who can afford it. And I think even in countries that have a more civilized approach to healthcare, probably IVG is not going to be high on the list of things that the National Health Service is going to be eager to pay for. My guess. I don't know your system very well, but it's a guess. So, last set of issues. And this is what the book went into in great detail. If IVG works, let's say you're a couple who wants to have babies, and you don't want to have unibabies or multiplex babies. You just want to have normal, everyday babies. You don't need to use IVF. But if you use IVG to get the eggs, you avoid egg harvest, which cuts out a lot of the cost and a lot of the unpleasantness and the risk. IVF becomes easy. If you do IVF, by the way, how many embryos can you make? How many eggs do you get? Well, with regular egg harvest with a healthy young woman, you get, 0 to 25, usually around 10 to 15. If you're making them from a cell line, from a induced pluripotent stem cell line, how many eggs do you get? How many do you want? Hundreds, thousands, millions. So in the book, I use as a hypothetical, you make 100 embryos. You do a whole genome sequencing on each of those 100 embryos. And you ask the prospective parents, which one or two do you want us to try to transfer? It takes embryo selection to a much higher level. And I think that actually could happen. In the book, I predicted will happen and will be relatively common, not for enhancement purposes. We don't know enough about genes for enhancement to do any real significant enhancement, but for health purposes. About 1 to 2% of every birth is the birth of a baby who has a serious or will have a serious disease that could have been predicted through prenatal genetic testing. If you could avoid that, you're avoiding human suffering for the baby and the family. You're also avoiding a lot of healthcare costs. I think health insurers and health plans, health programs might be quite interested in paying for IVG if it meant they didn't have to pay as much in taking care of children who had serious genetic diseases. So I think that may be where we eventually end up. IVG, as you can see, is something I find deeply interesting and think has lots of consequences. Last point, what should we do? First, we should watch it carefully. Second, I think we should fund the research. I mean, frankly, I see no good argument against IVG, apart from safety, always important. But carefully proven safe IVG for people who are agametously infertile, and for me, that would include gay and lesbian couples. I think there's no good argument against it. There will be arguments against it, but I don't think there are good arguments against it. I would fund research toward that. I would engage with the public. I would focus very heavily on what kind of proof of safety you would require, and then start deciding what your country wants to allow and wants not to allow. Different countries will make different decisions. Everybody should ban involuntary reproduction. Everybody should ban stealing Andrew's cells to make him a dad without his knowledge. That shouldn't be legal. Although there may be some exceptions. If Andrew gets hit by a lorry and he and somebody had been planning to have babies, he's dead, he can't give consent, maybe there's a posthumous exception there. That's, I think, one of the easy ones. Different countries will differ. They need to start thinking about it. So I hope in 30 minutes, I have given you an introduction. Go buy, The End of Sex. It's not as scary as its title. It's on Kindle. It's like probably £7, of which I would get at least 30 pence. So thanks for your time and attention. And I'm looking forward to those royalties just rolling in because I've yet to make the minimum wage on that book. Thank you.
Thank you very much, Hank. That's,
You're still obviously flustered by the thought of being an involuntary dad.
I'm not buying any more coffee in paper cups.
Gobsmacked.
Well, thank you very much. That was tremendous.